Hello students and welcome to the course 21SM AC234B, that is the analysis of controlled substances. In this video we are going to discuss the analysis of diamorphine and heroin. The heroin, which is known by various street names such as dope, junk and smack, is a generic term that describes a group of compounds which have been made during the preparation of diamorphine from the morphine, which is extracted from the latex of the field poppy (Papaver somniferum). Pure heroin or diacetylmorphine or dimorphine is a white powder with a bitter taste abused for its euphoric effects. Heroin was first synthesized legally from morphine in the late 1800s. It is usually injected, smoked, or snorted up the nose. It exhibits euphoric, anti-anxiety, and pain-relieving properties. Because abusers do not know the actual strength of the drug or its true content, they are at often risk of overdose or death. The effects of overdose may include slow and shallow breathing, hypotension, blue lips, nails, muscle spasm, convulsions, coma, and possible death. Intravenous use is complicated by other issues such as the sharing of contaminated needles, the spread of HIV or AIDS, hepatitis, and toxic reactions to impurities. Other medical complications of using heroin may arise, and it includes collapsed veins, spontaneous abortion, endocarditis, pneumonia, addiction, etc. Heroin is derived from morphine, which is obtained from the latex of the field poppy (Papaver somniferum). As with all drugs, the sites of major production change with time. In 1995, it was estimated that the world opium harvest was 4,157 tons, with the majority being produced in Southeast Asia, especially Myanmar, Thailand, China, Vietnam, and Cambodia. More recently, Afghanistan has emerged as a leader of opium production, producing over 4,500 tons in 1999, with the Southeast Asian production dropping by about 50%. In addition, small-scale production occurs in the Central and Southern America, with Colombia producing about 60 to 70 tons and Mexico producing about 50 tons per annum. The dimorphine is prepared by the isolation and acetylation of morphine. While the morphine was first discovered in the dried latex of Papaver somniferum shortly after 1800s, it is now known that of the approximately 110 species of Papaver family, only two are known to produce morphine in significant quantities. And these two plants are Papaver somniferum and Papaver setigerum. Of the two species, it is Papaver somniferum which is used to provide the dried latex for heroin production. Morphine is extracted from the dried latex from the seed capsules. Incisions about 1 millimeter deep are made in the seed capsule approximately 2 weeks after the petals have fallen from the flower. and the seed head is mature, and the latex exudes from the seed head, dries and oxidizes in the sun, and is collected the following day. With successive harvestings, a pod may produce anywhere between 10 to 100 milligrams of opium. The latex contains 10 to 20 weight% alkaloids, with the remainder of the latex being sugars, proteins, lipids, gums, and water. The opium contains morphine, codeine, and thebaine. In order that the morphine can be converted to diamorphine, it is necessary to extract and purify the morphine from the opium and synthesize and purify the diamorphine. In order to extract the morphine, the opium resins must first be prepared, and these are prepared by adding the raw opium to boiling water in which the alkaloids dissolve. While the insoluble material can be removed when it floats or it is filtered from the solution. To the extract the morphine from the processed opium, the filtrate is placed in a large volume of boiling water and calcium hydroxide is added. Then the water is cooled and the unwanted alkaloids precipitate, while the morphine and some codeine remain in the solution. The solution is then reheated and ammonium chloride is added. When the pH reaches to 8 to 9, the mixture is cooled and the morphine and any remaining codeine precipitate out. The crude morphine is dried in the sun, ready for the next stage in the process of synthesis. The dimorphine is prepared from the morphine, and the synthesis is achieved by mixing the morphine with acetic anhydride and heating to approximately 85 degrees Celsius for about 5 hours or until all of the morphine has dissolved. Then the water is added to the mixture, followed by the activated charcoal, and the mixture is repeatedly extracted with the charcoal and filtered until the solution is clear. Sodium carbonate dissolved in hot water is slowly added to the mixture, and the heroin base precipitates as a solid, which is then filtered and dried. For each kilogram of the morphine, up to 700 grams of diamorphine can be produced from this procedure. While when we talk about the bulk and trace sampling procedures, if the samples can be easily contaminated, we call it as a trace sample, otherwise it will be treated as a bulk sample. In general, the material should be separated out into visually identical groups, and the number of atoms in each group should then be determined. One model proposed by United Nations Drug Control Program suggests that if the group size is less than 10, then each member of the group should be sampled. If the size is between 10 and 100 packages, then 10 packages from the group should be sampled. And if the size is greater than 100 atoms, then the square root of the number of atoms should be sampled. The atoms to be identified for sampling should be chosen randomly, and the selected samples or the chosen sample can be subsequently identified, quantified, and compared with the heroin samples. Whatever the type of the sample Having carried out a description of the material and obtained a suitable sample for analysis, the next stage is the analytical process itself. That is, to identify, quantify, and when necessary, make the comparison between the samples. If the sample is a trace sample suspected of being heroin, the usual route is directly to GCMS analysis. And if the sample is a bulk sample, the analytical sequence includes presumptive tests, TLC, confirmation of identity usually by GCMS, and quantification and further sample comparison by HPLC if required. There are number of presumptive tests are used to identify the heroin from the sample. The opiate drugs can be screened for heroin by using a combination of tests as shown in table 5.1. Each of the tests alone are not specific to one opiate drug. This means that further tests are required to determine which of the opiates are present. For example, the Marquis test, the opiates react to give blue or to violet products on a colorless background. In Mendelian reagent test, the opiates react to give olive green products on an orange background. Whereas in cobalt isothianate test, the diamorphine reacts to give a blue product on a pink background and the other opiates do not react. So this cobalt isothianate can be exclusively used for the identification of the heroin from the sample. The thin layer chromatography is followed by the presumptive test. After the presumptive identification of the materials as potentially containing opiates, the next phase of the analytical sequence is to confirm the identity of which opiates are present and to carry out a comparison of the samples. The samples for thin layer chromatography should be dissolved in a solvent and the solution then applied to the TLC plates. Of the solvents available, chloroform has been used for heroin analysis. The samples should be dissolved at a concentration which is of the same as the standard against which they are to be compared. The TLC of heroin sample is most frequently carried out on silica gel. Between 5 and 10 microgram of each component drug should be applied to the plate. The solution should be applied in as small spot as possible and the positive control and the negative control should be applied to the chromatographic plates. After application of the mixtures, the plate is placed in a TLC developing tank which has been pre-saturated with solvent and then developed. There are a number of different solvent systems which can be used to separate the components of heroin mixture. And these different solvent systems and their separation is shown in the following table. For example, a methanol and ammonia mixture can be used to separate diamorphine, 6-O-monoacetylmorphine, morphine, and codeine efficiently, whereas the other adulterants can also be identified by using this solvent mixture. Whereas if cyclohexane, toluene, and diethylamine or chloroform and methanol are used, the adulterants are not identified in the TLC. After the development of the plate, the chromatographic plate is removed from the tank, dried at room temperature, and then visualized in the white light, short wavelength, and long wavelength light following the spraying with a suitable developing reagent. These spraying reagents may include acidified potassium iodoplatinate and Dragendorf's reagent. The data obtained after the TLC should be recorded at each stage, including the colors and the retardation factor or retardation value factor values of the compounds observed at each stage of the visualization process. Then the retardation factor of the sample and the standard is compared, it is matched. If the RF value and the color formed with this spraying reagent is matches with the standard, then the sample can be called to contain the heroin in it. Once the heroin is confirmed in the TLC analysis, then it can be further analyzed or identified by using GCMS and quantified by using HPLC, and if required the comparison with the other samples can also be done by using HPLC method. The procedure which is used for qualitative and quantitative analysis of heroin is same as which is used for other controlled substances, and the results can be expressed in terms of the presence or absence of heroin in the sample. The detailed procedure for GCMS analysis, its quantification and comparison, please follow the link in the description box.